Hello, let's discuss on applications of biotechnology in health and agriculture. What is biotechnology? Biotechnology is defined as the controlled use of biological agents such as microorganisms or cellular components for beneficial use. Biotechnology has enabled scientists to isolate a desired gene from organisms and then inserting them into other organisms to get a desired product or desired characters. Say, one organisms have tall height and the other organisms have a white complexion. When we want to have both the characters, that is tall height and white complexion, the gene or DNA from the tall height organisms can be extracted and then inserted artificially into the genome of an organism with the white complexion or vice versa. So after insertion, the resulting organisms will have both the characters, that is tall height and white complexion. That means the genome of the organisms with white complexion is also containing the functional foreign genes or DNA from the tall height organisms. The gene also the white complexion organisms is also containing the functional foreign gene or DNA of the tall height gene. So such organisms whose genome contains functional foreign genes or genes introduced artificially by the genetically engineered by genetical engineering are called as transgenic organisms or transgenic. The inserted foreign gene from the unrelated organisms is referred to as transgene, and the science that is dealing with the production of transgenic organisms is called as transgenesis or transgenic technology or genetic engineering. Transgenic organisms are also called as genetically engineered organisms or genetically modified organisms, that is GMO. The transgenic organisms can be microbes, plants, animals, or even fungus. Now we shall discuss on the development of transgenic organisms. There are different techniques used for introducing foreign DNA into the host cell. The first technique is by using a vector as a vehicle for gene transfer. Now vector here, in simple terms, we are referring to the organisms that is carrying the foreign gene. And let's take an example of mosquito. Mosquito used to transmit the malarial parasites into human body. So here mosquito is referred to as the carrier of the malarial parasite. That means mosquito is a vector, and the living organisms to whom the malarial parasite is being transmitted, that organisms is referred to as host. So using vector as vehicle for gene transfer, the DNA segment from the desired organisms can be isolated and then inserted into the DNA of a plasmid phage or virus, and this DNA of the plasmid phage or virus can be used as a vector for introducing the gene of interest into the host organisms. The second method of transfer of this the second method for developing these transgenic organisms is by vectorless or direct introduction of foreign gene. Now in direct gene transfer, the foreign genes of interest is directly delivered into the host cell without the help of any vector. So some of the techniques used in this vectorless method are chemical mediated gene transfer. In this chemical mediated gene transfer, presence of certain chemicals like polyethylene glycol are required. The second technique is by electroporation. In this electroporation, electric impulse with high voltage are applied across the protoplasm of the cell, and when these electric impulse are introduced into the protoplasm, a temporary pores across the plasma membrane are produced. So this temporary pore is used as a space for introducing the foreign gene into the cell. The third technique is by particle gun method. In this method, foreign DNA is coated into the surface of a minute particle like gold or tungsten and is fired with high speed into the target cells. Now we shall discuss on the applications of biotechnology in healthcare. Biotechnology is obvious have many great advantages in the commercial production of genetically engineered products, and for diagnosis of various type of diseases. First, let's discuss on therapeutic drugs or pharmaceutical product. Under this pharmaceutical or therapeutic drug, the first one will be genetically engineered human insulin. Insulin is a hormone that is used to treat the diabetes mellitus. Earlier, the insulin hormone was extracted from the pancreas of the slaughtered animal like pig or cattle. However, the insulin from these animals are found to produce certain type of immunological response, and hence the first human insulin was produced from bacteria named Escherichia coli or E. coli in 1983, and this first human insulin was named as humulin. Human insulin is formed of 51 amino acid chain organized into two polypeptide chain. So the two polypeptide chain are A chain or alpha chain and B chain or beta chain. These two polypeptide chain are synthesized together as a single polypeptide chain called as the proinsulin, and it also contains extra additional stretch of amino acid called as the C peptide or connecting peptide. The proinsulin, however, needs to be processed before it becomes fully matured and functional as an insulin hormone. So during its processing, the C peptide is removed from the two strands of the polypeptide chain, that is alpha and beta chain, with the help of an enzyme such as endopeptidase. So once the C peptide is being removed from these two strains of the polypeptide chain, this C polypeptide chain also induces the formation of disulfide bond between the two strains, that is A and B chain of the amino acids, and this formation of disulfide bond helps in the connection between the alpha and beta chain. So once this alpha and beta chain is being connected, it becomes a fully functional insulin hormone. Now we shall be discussing on the C peptide. What is C peptide or connecting peptide? C peptide or connecting peptide is a protein containing around 31 amino acids chain, and it also remain connected to the alpha or beta chains of the insulin in the proinsulin molecule. The C peptide chain helps in the formation of disulfide bond between the alpha and beta chain of the insulin molecule, and the presence of this C peptide chain also help in the diagnosis of diabetes disease. Under therapeutic disease and under therapeutic drugs or pharmaceutical product, the second is the medically important protein. Biotechnology also help in production of two important medical proteins such as tissue plasminogen activator and atrial peptides. Out of these two, tissue plasminogen activator is a human protein that has been synthesized for dissolving blood clots, and this protein is also found to be effective in preventing heart attack and stroke. The second protein, that is atrial peptides, also helped in treating blood pressure and kidney failure. Now we should be discussing on the gene therapy. What is gene therapy? Gene therapy involves the treatments of the hereditary disorder that is caused by the single defective genes. So in gene therapy, the single defective gene is being replaced by a normal genes. The first gene therapy was tried on a girl who is suffering from disease called as the adenosine deaminase deficiency. So this deficiency is also called as the severe combined immunodeficiency or SCID. So adenosine deaminase deficiency, also called ADA deficiency, is a rare genetic disorder of immune system, and it is autosomal recessive disease caused due to the loss or deletion of the gene that is coding for enzyme adenosine deaminase. Now during the gene therapy of this ADA deficiency disease, the lymphocytes or the white blood cells from the patient blood are grown into the culture medium outside the body of the patient, and a functional adenosine deaminase complementary DNA is introduced into the lymphocytes using retrovirus vector. So after introducing this ADA complementary DNA, the lymphocytes is then introduced or retransferred or returned into the patient's blood. However, once the genetically engineered lymphocytes containing the ADA complementary DNA is introduced into the blood of the patient, it becomes integral part of the lymphocytes and begin to mature from the bone marrow cells. However, the patient needs required periodic infusions of the genetically engineered lymphocyte cell, because these lymphocyte cells are not immortal and they die out once their lifespan is over. So this treatment is only temporary. So in order to achieve a permanent cure from this ADA, the genes isolated from the normal bone marrow from a normal persons have to be introduced into the cell of the patient at the early embryonic stage. So this is all about the gene therapy.